Hello everyone, welcome to my YouTube channel. I hope you all are fine. So last time we discussed the neuromuscular junction, and in today's video we will talk about the synaptic transmission. So we have two types of arrangements. The one-to-one synapse that is found at the neuromuscular junction that we have discussed in the previous video, that an action potential in the presynaptic element, that is the motor nerve, it produces an action potential in the postsynaptic element, that is the muscle. So we have one presynaptic element and one postsynaptic element. So this is the one-to-one synapse, and another one is the many-to-one synapse, and that is found in the spinal motor neurons. So an action potential in a single presynaptic cell, it is insufficient to produce an action potential in the postsynaptic cell. So what happens that many cells, they synapse on the postsynaptic cell to depolarize it to the threshold. And the presynaptic input, it may be excitatory or inhibitory. So many presynaptic cells, they synapse onto a single postsynaptic cell to depolarize it and to generate the action potential. So this is many-to-one synapse that is found in the spinal motor neurons. Okay, and the next topic is the input to synapses. So in the previous video, we talked about that the postsynaptic cell integrates the excitatory and inhibitory inputs. So the input is coming from the presynaptic cell, and that may be excitatory or inhibitory. So the postsynaptic cell integrates the excitatory and inhibitory inputs. So these inputs, they summate, and then they bring the membrane potential of the postsynaptic cell to the threshold. So when the sum of the input brings the membrane potential of the postsynaptic cell to threshold, it fires an action potential. Okay, so the excitatory postsynaptic potentials, they depolarize the postsynaptic cell. Like in the previous video, we talked about the neuromuscular junction, and the input that is coming from the presynaptic cell, it depolarizes the postsynaptic cell and bringing it close to threshold and closer to firing the action potential. And this is caused by the opening of channels that are permeable to sodium and potassium, similar to the acetylcholine channels. And the membrane potential depolarizes to a value halfway between the equilibrium potentials for sodium and potassium, approximately 0 millivolt. And what are the excitatory neurotransmitters? This includes acetylcholine, norepinephrine, epinephrine, dopamine, glutamate, and serotonin. Okay, then the inhibitory postsynaptic potentials, these are the inputs that hyperpolarize the postsynaptic cell and moving it away from the threshold and farther from firing an action potential. And this is caused by the opening of chloride channels. Now the membrane potential is hyperpolarized towards the chloride equilibrium potential, that is minus 90 millivolt. And what are the inhibitory neurotransmitters? These are the gamma-aminobutyric acid, GABA, and glycine. Okay, that's just a summary of what is written in the BRS Physiology. For excitatory postsynaptic potentials, remember that these are the inputs that depolarize the postsynaptic cell. Inhibitory postsynaptic potentials, they will hyperpolarize the postsynaptic cell. And for the excitatory postsynaptic potentials, there is opening of channels that will increase the permeability of sodium and potassium. And in case of inhibitory postsynaptic potentials, there is increased permeability for the chloride ions. And also remember the examples of neurotransmitters, excitatory and inhibitory. Okay, and the last topic on this page is the summation at the synapses. So we have two types of summation, the spatial summation and the temporal summation. Okay, so in the diagram, consider that spatial summation, like there are many people and they are working together. So in the BRS Physiology, it is written that the spatial summation occurs when two excitatory inputs, or we can say multiple inputs, they arrive at the postsynaptic neuron simultaneously, and together they produce greater depolarization. Whereas in the temporal summation, we have a single person doing all the work. So it occurs when two excitatory inputs, they arrive at the postsynaptic neuron in rapid succession. Because the resulting postsynaptic depolarization, it overlaps in time, so they add in the stepwise fashion. So remember that temporal summation, the action potential is generated by a single presynaptic neuron, and that is continuously firing the signals, and they are adding in a stepwise fashion. Whereas in the spatial summation, the action potential is generated by multiple presynaptic neurons simultaneously, and they together generate an action potential. And the last thing we have is the post-tetanic potentiation. So, tetanizing stimuli in the presynaptic neuron. It results in the increased postsynaptic potentials, and that lasts for a few minutes to hours. And that is actually caused by an increase in the calcium influx in the presynaptic neuron. And what happens that this increases the release of neurotransmitters. So we have a depolarization of the postsynaptic neuron that is greater than expected, because greater than normal amounts of neurotransmitters are released. And why? Because there is accumulation of calcium in the presynaptic terminal. Okay, that's it for today's video. In the next video, we will talk about the different types of neurotransmitters. Like, share, and subscribe to my channel. I'll see you in the next video.